اهلا بيكم ابنائنا وبناتنا طلاب وطلبة الثانوية العامة في حلقة جديدة لمادة البيولوجي. هنعمل في خلال ساعة عدد من الأسئلة المهمة في البيولوجي ونشوف لو عرفنا نجاوب على كل الأسئلة صح هيبقى احنا مستوانا جميل وممتاز. لو في أسئلة وقفت معانا يبقى في نقط عايزين نعمل عليها ستريس شوية. نبدأ كده بأول سؤال. بيقولك choose the correct answer. In the human female the primary oocyte is formed. The first choice is a before the puberty. Number B after puberty. Number E during fertilization. Number D during the embryonic stages. The question asks about the primary oocyte or the formation of the primary oocyte. Of course, the right answer is during the embryonic stages. The second question. Which of the following female menstrual cycle disappear? Number A, multiple 6, 44 plus triple X or more than triple X. Number B, Turner syndrome, 44 plus X. Number C, Down syndrome, 45 plus XX. Number D, normal female, 44XX after puberty. The right answer is Turner syndrome, because Turner syndrome chromosomal structure is the 44X with one X chromosome only. So the Turner female can't produce sufficient amount of female sex hormone to reach the sexual maturity. So there is no menstruation cycle of the Turner syndrome female, because this female can't reach the sexual maturity due to the presence of one X chromosome only, which is not enough to reach sexual maturity. So the second question. The opposite diagram represents the unit structure of DNA, but it does not represent the unit structure of RNA when? Number A, the structure C represents phosphate group. The structure C represents phosphate group in both of DNA and RNA. The structure B represents ribose sugar. That's not right answer, because the ribose sugar is the sugar of RNA. But in the case of DNA, the B is the deoxyribose sugar. Number C, the structure A represents adenine. The structure A represents adenine in both of DNA and RNA. But the last answer, number D, the structure A represents thymine. That is not represent the unit structure of RNA, because RNA has no thymine but containing uracil instead of thymine. The next question. Starfish reproduce asexually by dots, budding, spores, regeneration, binary fission. The right answer is regeneration, because if you cut the starfish into many pieces, each piece pass through a certain part of the buccal cavity or the buccal disc that allow the part which containing the buccal disc or part of the buccal disc develop to produce complete starfish that act as the kind of asexual reproduction, which is called the asexual reproduction by regeneration. The next question. Amino acids are linked with each other in the polypeptide chain by organic nitrogenous bases, peptide nitrogenous bases or peptide bond, number C, phosphate group, number D, hydrogen bond. That's very simple answer, because the amino acids are linked together by peptide bond by removing one water molecule between the two amino acids during the formation of dipeptide. The next question. Single ring nitrogenous base which paired with the complementary nucleotide by 3 hydrogen bonds in the DNA helix is? The question asks about the nitrogenous base which paired with the complementary nucleotide, but that's single ring. The single rings are thymine and cytosine. Thymine paired with adenine by double hydrogen bonds, but the question asks about single ring paired with hydrogen bond by 3 by the nitrogenous base by 3 hydrogen bonds. That is the cytosine, because cytosine made of single ring and paired with the guanine by triple hydrogen bonds. The next question. The formation of the ciliated sperms in polypodium plant takes place in the archegonia, number A, number B, in the sporogonia, the number 3, the bimitotic division, the last answer by meiotic division. The formation of most of the gametes or reproductive cells in the higher living organisms takes place by meiosis. But in the case of the polypodium, the gametophyte is formed of haploid cells, and the haploid cells can't reproduce by meiotic division. So the division during the formation of the sperms or the gametes in the polypodium takes place by mitosis division. Not takes place inside the archegonia, because inside the archegonia the XL or the female gamete is formed. The next question. When Mendel carried out crossing between homozygous green seeds pea plant with the homozygous yellow seeds, they produce a generation with 100% green seeds, number B 100% yellow seeds, number C 50% green and 50% yellow. The last answer or the last choice is 75% green, 25% yellow. So the answer or the correct answer is 100% yellow seeds, because we know that the yellow seeds in pea plant dominant over the green. And Mendel explained during the Mendel first law or the law of segregation that when two homozygous individuals are different in a pair of allelomorphic characters are crossed, the dominant character appear in the F1, while the two characters appear in the F2 in the ratio of 3 dominant to 1 recessive. So the dominant character appear in 100% in F1, and in this case the yellow seeds is dominant over the green. The next question. The correct sequence of nitrogen nucleotides that exist in the DNA strand of the glycine amino acids, as shown in the opposite figure, is dots. The first choice is GGA, B CCT, C GGT, D CCA. This part of the image act as or represent the tRNA. This glycine amino acid is attached to the attachment site of amino acid as a CCA at the 3' end of the tRNA. But the CCA act as an anticodon, and we remember that the anticodon is the 3 successive nucleotides on tRNA paired with the 3 codons on the mRNA, which represent the amino acid. So if the anticodon CCA, that means the codon on the mRNA is the GGU. So that means the nucleotides or the sequence of nucleotides in DNA is the CCA, because CCA during the transcription by RNA polymerase change into the codon GGU on the mRNA. And the anticodon of the GGU on the mRNA is the CCA. So the correct sequence of nucleotides that exist in the DNA strand of the glycine amino acids that shown on the opposite figure is the CCA. The next question. Compare between 6 linked and 6 influenced characters. We know that in the case of the 6 influenced characters, the genes of these traits are carried on an autosome or on the autosomes. But the 6 linked, the genes are carried on the 6 chromosome X. The action of these genes is influenced by the presence of hormones. But the action of these genes in the case of the 6 linked character is not affected by the hormones in the case of 6 linked character. The characters may transfer from the father to his sons in the case of the influenced character, because the son get the autosomes from the father. But in the case of the 6 linked character, the character can't transfer from the father to the sons, because the sons get the Y chromosome from the father, not the X, which carry the 6 or act which carries a sex-linked gene. The gametophyte of the impure individual appear with the two phenotypes according to the sex, as a B positive B in the male that act as a polled male, in the female act as a normal female, so that one genotype represents two phenotypes. But in the case of the sex-linked character, the genotype of impure individual, that's dominant character only. In the case of the sex-influenced character, the heterozygous may be male or female, but the male's pure in the case of the sex-linked character, so the heterozygous individuals, females only. The example of sex-influenced character is the premature baldness. The examples of the sex-linked character is eye color of Drosophila fly, color blindness, and hemophilia in men, and also the muscles atrophy in men. So that's the comparison between the sex-influenced and sex-linked characters. The next question. The genetic problem in Drosophila fly, the red color of eye is a dominant character. A red color or the eye color of Drosophila fly, the red and the white color of Drosophila fly represent the sex-linked character. And it's linked to sex. That means the gene is linked on the X chromosome. On genetic basis shows the result of the crossing between heterozygous red-eye female and number A, white-eyed male, number B, red-eyed females. In the case of the sex-linked character, the females represented by XX and the male represented by XY in the case of the human, Drosophila fly, and the mammals. But the opposite takes place in the butterfly, birds, and some fish. So, the solving of the problem, the first generation is a male with the white eyes, the female, that means the X carry R small letter with the Y. The female XX are capital R small. Segregation of the gametes of the male, one of them X containing or carry R small and the other sperm with Y. But in the case of the female, two kinds of gametes, X carry R capital and the other X carry R small. So the F1 will be 50% red and 50% white. But in the case of the red-eyed male and impure red female, that will form, or the two parents will be X R capital Y and X R capital X R small. So the ratio in the F2 will be 3 red to 1 white, but the white is male because that act as a sex-linked character. The next question. Give scientific term for each of the following. A deep depression at the connection between the ilium and ischium bones. The depression is the acetipulum cavity where the head of the femur fit inside it to form the thigh joint. So we remember that the two cavities of the upper limbs and the lower limbs, or the forelimbs and the hind limbs, the upper limbs which is represented by the humerus bone, the head of the humerus bone, fit in the glenoid cavity of the scapula. But the head of the femur of the lower limb fit in the acetipulum cavity in the pelvic bones. The next, give scientific term. Cells are produced from the meiotic division of the spore mother cell in the anther. The spore mother cell in the anther is divided by meiosis to produce 4 cells. Each is called microspore. So the answer is microspores. The microspores are 4 haploid cells produced as a result of meiotic division of the spore mother cell in the anther. The next question, give scientific term. A strain of bacteria causes pneumonia to mice and doesn't kill them. We remember that there are 2 types of pneumonia bacteria which relates to or causing pneumonia disease. One of them is called the virulent bacteria, which is causing a disease and leads to this, but the other is non-virulent bacteria. The non-virulent bacteria, which act as the rough bacteria, so that represented by R capital, that's causing pneumonia disease but not leads to this. The question asks about a certain type of bacteria which is causing pneumonia disease but doesn't kill the rats. The answer is non-virulent pneumonia bacteria. Non-virulent means a bacteria which doesn't cause death of the diseased individual. The next question. The largest bone of the 7 tarsals that form the ankle. The ankle bones consists of 7 bones. The largest one is the heel bone. So the answer is the heel bone, which acts as the largest bone in the 7 tarsals that form the ankle of the human. The next question. The fusion of 2 male nuclei, one with egg and the other with the embryo sac nuclei. That's double fertilization, because the male gamete which act as the pollen grain containing two generative nuclei, or two cells. One of them is the generative nucleus and the other is the pollen tube nucleus. The generative nucleus is divided by mitosis into 2 male nuclei. One of them is fused with the egg cell to form the zygote, and the second one will fuse with the 2 embryo sac nuclei to form endosperm. The endosperm acts as the food of the embryo. So the fertilization takes place by 1 pollen grain is the fertilization of egg cell and the 2 embryo sac nuclei. So that's called the double fertilization. But the fusion between one of the 2 male nuclei with the 2 embryo sac nuclei to form endosperm only, that act as the triple fusion. So the double fertilization act as the formation of zygote and the triple fusion to form the endosperm. The next question. The enzymes recognize a certain site on the DNA molecule and cut it at these sites. The enzyme which recognizes certain parts of DNA or certain sites on DNA and cut it into pieces or inclined sticky ends, that's called the restriction endonuclease enzyme or the restriction enzyme. So the restriction enzyme cuts the DNA in a certain parts or with the 2 inclined sticky ends at a certain sites. We remember that the bacteria protect its DNA against the action of its restriction enzyme by the presence of the modification enzymes which put methyl group on the recognition site. The next scientific term. Human genes that located on the chromosome number 8. The answer is the fingerprint gene. So the gene of the fingerprint is located on the chromosome number 8. The next question. The fruit in which the receptacle or any part of the flower sharing the ovary to store food, as that in apple. The fruits which containing any part of the fruit sharing the ovary to form fruit is called the false fruit. So the right answer is the false fruit, as that of the apple. The next question. Small circular DNA molecule found in some bacteria cells. The small circular DNA is a plasmid, which are located in the prokaryotes. The plasmid is used in many steps of the genetic engineering. The next question. The ability of the ovary to develop into seedless fruit by using the auxin. The ability of the ovary to change into fruit without pollination or fertilization. That's the same question. So the answer is the artificial parthenocarpy. We remember that the parthenocarpy is the ability of the ovary of the flower to change into fruit without pollination or fertilization. And there are 2 types of parthenocarpy. One of them is called the natural parthenocarpy, that takes place without the interference of the human beings, as that of the banana, but the other artificial by using the auxin as the indole and naphthol acetic acid, which stimulate the ovary to grow to form fruit without seeds. So that's called the seedless fruit that takes place by the artificial parthenocarpy, as that in the cucumber and the tomato. So the artificial parthenocarpy is not act as a kind of reproduction. It just act as the growth of the ovary to change into fruit, but the fruit doesn't containing seeds. So that's not act as a kind of reproduction, but it just act as the growth. The next question. Give reason for. Wheat is called grain, but beans is called seeds. Wheat is a grain, but bean is a seeds. We remember that in the case of the grains, any type of grain, the two integuments and the ovule, or the ovule, the ovary wall, all of them are fused together to forming a single-walled the seed fruit. So that's called the grain. But in the case of the bean, the two integuments of the ovule hardening, forming a testa, while the ovary wall change into precarp. So that's act as a seed. So the seeds are located inside the fruit, and each seed is surrounded by the testa, which act as the two integuments which surround the embryossac. But in the case of the grain act as a single-walled fruit due to the fusion of the integuments with the ovary and the ovary wall together to form the grain. And that's the difference between the grain and seed. The next question. Muscle fiber need energy during muscle contraction. The muscle fiber need energy during the muscle contraction. Because the muscle contraction, or in the muscle contraction that takes place by pulling of the actin filaments as the two sides of the myosin filaments, that takes place by the transversal link that extended from the myosin filaments that need energy. So the pulling or the formation of the transverse links at the presence of the calcium ions and the ATP molecules that allow the two actin filaments as the two sides of the myosin of the myofibrils are pulled together inwards to shorten the length of the myofibrils that takes place during the contraction of the muscle. So the pulling movement need a certain type of energy, so that's act as a kind of the active transport, or takes place by active transport, which need energy. The next question. Give reason for. The seedless fruit can be obtained artificially. That's seedless fruit, that means the fruit without seeds. That's the artificial parthenocarpy, because that takes place by spraying the activating substance such as indole or nephsol acetic acid, or the extraction of the pollen grains that dissolve it in water or ether, so that spreads over the stigma of the flower, that stimulate the ovary to change into fruit without seeds. The next give reason question. Showering the body during the winter. Showering the body during winter. That takes place to generate the sufficient amount of heat energy to keep our body temperature constant. So during the winter, the outer environment or the temperature of the outer environment decrease, so the body lose great amount of energy. So the showering that stimulates the body to generate sufficient amount of heat that to keep the body temperature constant. The next question. The human female reproductive system connected by elastic ligaments. The human female reproductive system is connected by elastic filaments or ligaments, the same, that act as a kind of connective tissue. The answer, that to allow the expansion of the female reproductive organs during the pregnancy. So this expansion needs the presence of elastic filaments that connect the female reproductive system together. The next question. Some flowers are called bisexual, while the others are called unisexual. We remember that bisexual means they carry the male and the female organs, but unisexual carries the male organs only. So in the case of the bisexual flower, the androsium and the gynosium exist in the same flower, which is called the bisexual flower. While the other flower, which are called the unisexual, that because the presence of one sex only, the androsium only or the gynosium only. So in the case of the unisexual flower, the presence of the male organs only or female organs only. But the hermaphrodite flower that containing the male and the female organs, which is called the bisexual. The next question, give reason. The testes are laid outside the body, or the testes are located in the scrotum, or the testes are located in the scrotal sac outside the body of the human male. The answer of these questions, because this position prepare a conditions cooler than the body temperature that give good chance of the spermatogenesis or the formation of sperms, because the sperms formation takes place at the temperature lower than the body temperature. But if the two testes are located inside the human body, that leads to the sterility or the infertility. And we remember that testes fail to descend down into the scrotum sac or the scrotal sac or the scrotum, that during the first embryonic stages or the embryo before the delivery. So the presence of these testes inside the human body that's containing a temperature higher than that suitable for the formation of the spermatogenesis. The next question. Formation of endospermic nucleus is called the triple fusion. The formation of endosperm nucleus that called the triple fusion. Triple fusion means 3N. 3N means fusion between the one of the two male nuclei of the pollen grain with the two embryo sac nuclei that form the endosperm, which act as the 3N cells. So that act as the triploid cells, which containing 3N, that act as a food to help the embryo to feed. And we remember that the seeds are divided according to the presence of endosperm into two types. The first is called endospermic seeds, which contain endosperm as that in the monocot, and the second is the ex-endospermic seeds, where the embryo consume all the amount of endosperm inside the seed, so that's called the ex-endospermic seeds. The ex-endospermic seeds are located in the dicot seeds, which containing two cotyledons, which act as a food stored inside the seed to allow the embryo to feed, because the embryo consume all the amount of endosperm inside the seed during the formation of the embryo. The next question, give reason. The embryo culture experiments is considered as asexual reproduction. The embryo culture experiments is considered as asexual reproduction. We remember that any kind of asexual reproduction takes place by single parent. So this process, which is called the embryo culture, it's completed by only one parent or one zygote to N, and all the embryos are identical in their characteristics, or they identical genetically, because we remember that the zygote is formed as a result of the fusion between one male nucleus and the one female nucleus. So the zygote is 2N. During the development of the zygote, or the zygote can divide it into two cells, which are called the plastomere after 1 day, and after that the division become faster than and to produce sufficient amount or great number of cells. If you separate the cells and put each part of the cells in the uterine female, that's allow the each part is developed to produce complete individual, which act as a which is called the embryo culture, that's act as a kind of a sexual reproduction, because it takes place by single parent. The next question. there are some fruits are called false fruit. We remember that the false fruit are fruit formed due to the sharing of certain part of the fruit with the receptacle to form the fruit, as that in the apple and fig. So in this case, or in the false fruit, that means the sharing of some part of the flower with the ovary to form fruit. The next question. There are some twins are called Siamese twins. The Siamese twins are located in the case of the identical twins. And we remember that the identical twins is formed by the fertilization of one ovum with one sperm. That the cells or the zygote develop to produce the embryonic cells. May these cells separate to form two masses of cells, each develop to produce two embryos or two fetus, but may some of them adhere each other or attach with each other in a certain part, so that's called the Siamese twins. So the Siamese twins are identical twins formed by the fusion of one ovum with one sperm. But because of the presence of the two embryos in the same embryo sac, may one of them attach of the other in a certain part of the body. So the two fetus that attach to each other in a certain part of the body, that's called the Siamese twins. The next question. What's meant by muscle bundle? The question asks about the muscle bundle. Muscle bundle is a group of muscle fibers that connect together and surrounded by strong membrane, which is called the perimysium. So the muscle bundle is a group of muscle fibers connected together and surrounded by the perimysium. This image explain, or this video explain the muscle bundle. The muscle bundle is a group of myofibrils connected together to form a fiber, a group of muscle fiber connected together and surrounded by the perimysium, that is called the muscle bundle. A group of muscle bundles connected together to form the muscle, which act as a unit structure of the muscular system. The next question. What's meant by anticodon? The anticodon are three successive nucleotides on the tRNA that paired with the codon on the mRNA during protein synthesis or during the translation process. So the anticodon is the three successive nucleotides are located in the tRNA, which paired with the codon on the mRNA, which represent a certain amino acid. So that is the anticodon of the tRNA. The next question. What's meant by releasing factor? The releasing factor is a certain protein attached to the stop codon for the termination of the protein sense. Special kind of protein on the stop codon on the mRNA causing a termination of the protein sense. And we remember that the stop codons are UAG, UGA, and UAA. That act as the three successive nucleotides on the mRNA, where the presence of a certain protein, which is called the releasing factor, causing the termination of the protein sense by separation of the large ribosomal subunit and the small ribosomal subunit and the polypeptide chain spread in the cytoplasm, that leads to the termination of the protein sense that takes place only at the presence of the releasing factor, which is located on the stop codon of the mRNA. The next question. What's meant by interferon? Interferon is a kind of protein that decreases the ability of the virus genetic material to duplicate. That takes place when the human cells are infected by a virus. When the human cells infected by virus, a certain gene on the DNA or a number of genes on the DNA are translated into or transcripted into mRNA, and the mRNA translated into the certain sequence of polypeptide chains that form the interferon. The interferon is a certain protein that secreted by the infected cells and spread in the body, that to decrease the ability of the viral genetic material to duplicate and also protect the neighboring cells against the infection with the virus. So the interferon is used in treatment some viral disease as a hepatitis, and also in the treatment of some diseases as a cancer disease. The next question. The genetic problem. An interhinum plant, when two pink flowered plants were crossed, white and red flowered plant were produced. Explain this case on genetic basis. That crossing between interhinum plant or interhinum flowering plant that act as a lack of dominance case, because the flowers of the interhinums are pink, red, and white. So there is no gene dominant over the other. That means the dominant gene, or there is no gene dominant over the other, so the red represented by R capital, white represented by W capital. When the crossing takes place between pink, that means that's impure individual which carry two different genes. So the gametes are R capital, W capital. So the F1 which is formed as the result of this crossing is 1 red, 2 pink, and 1 white. Lack of dominance act as a genetic case, which represented by 3 phenotypes in the genetic problem, and also the ratio of the F2 is a 1 to 2 to 1, because if crossing takes place between red and white flower, red represented by R capital, white by W capital, the pink which is formed as a result of the fusion between the two genes R capital, W capital, that form an intermediate character. So there is no gene dominant over the other, but each of them sharing with the same strengths to form the character. So the two intermediate genes, or the intermediate character, if crossing takes place between two pink flowers, that will form the 3 phenotypes, red, pink, and white, with a ratio 1 to 2 to 1. The next question. Show on genetic basis the ratio of the offspring from the crossing between a heterozygous black male mice with the brown female. Black and brown. Which of them act as a dominant character? Black is a dominant character because the black is a heterozygous individual. The dominant character may be pure or impure, but the recessive character is pure only. So the black male represented by B capital P small, but the brown represented by P small P small. So the gametes is the separation or segregation of the P capital and P small, but the female gamete is one gamete is a P small. The F1 formed by the fusion between the sperm and the ovum that form B capital B small and the P small P small. So the gametophyte is a B capital B small. The genotype is impure in the first one, but pure in the case of the recessive one. The phenotype is a black and brown, but the ratio 1 to 1. So this case act as a complete dominance, because the black gene complete dominant over the brown one. The next question. Compare between DNA in prokaryotes and the DNA in eukaryotes. In the case of the DNA in prokaryotes, it's surrounded by the nuclear membrane due to the presence of nucleus in the prokaryotes. But there is no nuclear membrane in the case of the eukaryotes. In some of the, or in the case of the prokaryotes, the DNA appear in a circular shape. But the DNA in the eukaryotes consists of double-stranded with 2 free ends. In the case of the prokaryotes, DNA is not complexed with protein, but in the case of eukaryotes, DNA complexed with protein to form a nucleosome or to become chromosomes. In the prokaryotes, the DNA is attached to the cell membrane at a certain attachment point where duplication takes place. But in the case of the eukaryotes, duplication takes place at any point. In the prokaryotes, the presence of a small circular DNA, which is called the plasmid, but no plasmid is located in the eukaryotic cells. In the case of the prokaryotes, a single DNA polymerase is used to transcribe 3 types of RNA. But in the case of the eukaryotes, 3 types of RNA polymerase. One of them to form tRNA, and the second to form mRNA, and the third to form rRNA. Because of the presence of 3 types of RNA, so 3 RNA polymerase are present in the case of the eukaryotes, but in prokaryotes, single RNA polymerase to form the 3 types of RNA. In the case of the prokaryotes, transcription and translation takes place at the same time in cytoplasm. But in the case of the eukaryotes, transcription takes place inside the nucleus. After that, translation takes place in cytoplasm. The examples of prokaryotes is a bacteria. The example of eukaryotes is man. So the difference between the bacteria and the other higher living organisms, or the prokaryotes and the eukaryotes, that's the difference between, or in the case, in the structure of the genetic material in both of them. The next question. Compare between the releasing factor and the recognition site. The releasing factor is the special protein on the stop codon on the mRNA causing termination of the protein sense. But in the case of the regeneration, the recognition site, in the case of the recognition site, specific sequence of 4 to 7 nucleotides where the restriction enzyme snaps DNA or cut DNA at or near these sites when the 2 inclined sticky ends formed. So that means the restriction endonuclease enzyme cut DNA in a certain piece which is called the recognition site. In this site, the restriction enzyme cut DNA into pieces with 2 inclined sticky ends. So that's very important during the genetic engineering, that to join a certain gene with a certain plasmid to produce plasmid carrier genes that introduce inside the cell to form a certain protein that produced by this gene. That as in the case of the formation of insulin, by carrying the gene of insulin on the plasmid and introduce the plasmid which carries gene inside the bacterium cell. We remember that all the genes of the bacteria are codons, or the genomes, most of them is codon, so the gene is translated into the corresponding protein that in the case of the formation of human insulin or human interferon or the human hemoglobin, in the case of the carrying the mRNA from the red blood cell to change it into DNA, and the DNA duplicated by the DNA polymerase to form the gene which carried on the plasmid. The next question. Compare between the codon and the anticodon. The codon is 3 successive nucleotides on mRNA represent a certain amino acid, or represent stop codon during the protein sense. But the anticodon is the 3 successive nucleotides on tRNA, are paired with the codon in the mRNA and represent a certain amino acids during protein sense. So the anticodon on the tRNA, but the codon on the mRNA. The anticodon is 3 successive nucleotides on the tRNA, but the codon 3 successive nucleotides on mRNA. The 3 successive nucleotides represent amino acids or stop codon in the mRNA. But the tRNA, or in the case of the tRNA, the anticodon represent a certain amino acids only. But there is no anticodon of the stop codons due to the presence of releasing factor on the stop codon on the mRNA which terminate the protein sense. But there is no tRNA on the stop codon because the stop codon is not represent amino acid, but it carry the releasing factor for the termination of the protein sense. The next question. Study the opposite diagram which explain a part of the protein sense. Then answer the following question. Number A, indicate the name of the structures numbers 1 and 2. The structure number 1 is the amino acid or the first amino acid, which is the methionine amino acid. Number 2 is the anticodon of the tRNA, which carry or which may allow the tRNA to carry the certain amino acid. Number 4, the large ribosomal subunit. The part number 2 of the question explains the importance of the structure number 4. The structure number 4 is the large ribosomal subunit. We remember that the ribosome is formed by the large ribosomal subunit and the small ribosomal subunit, which are formed inside the nucleolus, and the 70 types of polypeptide chain formed in cytoplasm and pass through the nuclear pores to get inside the nucleolus to form the ribosome. Separation takes place of the small and large ribosomal subunit that spread in the cytoplasm, but they reunite again only at during the protein sense. So the large ribosomal subunit has two sites. P site means peptidal, A site means amino acyl site. And as a part of the ribosome, the presence of the 70 kind of polypeptide chains that form the enzyme. This enzyme is called the peptide transferase enzyme, which form a certain reaction which is called the peptide transfer reaction, that to connect the amino acids together by peptide bond. So by the connection of many amino acids together by peptide bonds during the peptide transfer reaction, that forms a polypeptide chain which is spread in cytoplasm during the termination of the protein sense by the action of the releasing factor. The part number 3 of the question indicate of the role that played by the codon 5 on the mRNA. The codon 5 is the UAG. UAG act as the stop codon. So the stop codon is the certain codon on the mRNA as the UAG, UGA, and UAA that carry the protein which is called the releasing factor, which is causing the termination of the protein sense, means the separation of the small ribosomal subunit, large ribosomal subunit, the polypeptide chain in cytoplasm. So that is the importance of the stop codon. The next question. Select from the column B what's suitable for the column A. Column A, number 1, shoulder girdle. Number 2, thoracic cage. Number 3, rib cage, or the rib only. Number 4, the pelvic girdle. But in the case of the column B, protect the brain. The part of the skeletal system that protect the brain is the skull, but there is no skull in the column A. The second point of the column B, where the humerus head fit. The humerus fit inside the glenoid cavity of the shoulder or of the scapula. The scapula containing a cavity which is called the glenoid cavity. So the humerus head fit in the glenoid cavity of the shoulder to form the shoulder joint. Protect the heart and the lungs. It is the rib cage or the thoracic cage which protects the heart and lungs against the shocks. Fix with the centrum of the vertebra from the back. The rib is connected or is fixed with two parts of the vertebra, the centrum and the transverse process. The last point of the column B, where the head of the femur fits. The femur or the head of the femur fit in the acetylum cavity of the pelvic girdle. So the answer of the column, the shoulder girdle with the humerus head fit. Thoracic cage protect the heart and lungs. Number 3, the rib, fixed with the centrum of the vertebra from the back. Number 4, the pelvic girdle where the head of the femur fit. But the last point, which is to protect the brain, is the skull, where there is no skull in the column A. So we remember that the skeletal system of the human beings consists of two parts. The first part is the axial skeleton, the second is appendicular. The axial skeleton consists of the skull, thoracic cage, and the vertebral column. The skull consists of two main parts. The first is a facial part, and the second is the cranial parts. The cranial part consists of 8 bones serrated with each other. But in the skull, the presence of a big hole, which is called the foramen hole. The foramen hole, it is the hole which allows the connection between the brain and the spinal cord. That's the first part of the axial skeleton, which is called the skull. But the second is the vertebral column. The vertebral column consists of 33 vertebrae. The upper 7 is the cervical, which are moderate in size. The next is the thoracic vertebra, 12, which are large in size. But the lumbar is the largest 5 vertebra below the thoracic vertebra. And the sacral vertebra are 5 fused together to form flat sacrum bone. The last 4 vertebrae are called the coccygeal vertebra that form the vestigial tail in the case of the human beings. That is the vertebral column. But the thoracic cage consists of 12 thoracic vertebrae, 12 pairs of ribs. The upper 10 pairs are connected as the dorsal side with the vertebral column, and at the terminal side with the sternum. But the whole 20 pairs of ribs are connected together. The whole 12 pairs of ribs are connected at the dorsal side with the vertebra in the certain part, which is the centrum and the transverse process. But the upper 10 pairs only are connected with the sternum bone. But the two lower ribs, number 11 and the pair number 12, are free at the terminal side, so they are called the floating tip ribs. But that is the axial skeleton, which is the skull, the vertebral column, and the thoracic cage. But in the case of the appendicular skeleton, which consists of the shoulder and the pelvic girdle. The shoulder consists of 2 identical halves. Each half consists of a scapula and the clavicle bone. In the scapula, the presence of glenoid cavity where the head of the humerus feet inside it. The humerus lower part is connected in a certain depression of the lower arm. The lower arm consists of radius and ulna. The ulna is a bone which is fixed, which act as a fixed bone, where the presence of the depression where the lower part of the humerus feet. But the radius move around the fixed ulna in a semicircular manner. At the end of the lower part, the presence of the carpals, which act as 8 bones in 2 rows. The metacarpals are 5 bones end with the fingers. Each finger consists of 3 phalanges except the thumb. But in the case of the pelvic girdle, that consists of 2 identical halves, each half consists of ilium, ischium, and the pubis, where the presence of the acetipulum cavity where the head of the femur fit inside it. The femur end with the knee joint containing the knee cap to allow the joint of the knee is a limited joint or limited joint during the movement. But the shank consists of the fibia and tibula. The fibia and the tibula each end with the foot of the human beings consists of the tarsals, metatarsals, and the end with toes. My dear students, that's the end of this episode. Thank you very much. Goodbye and good luck.